Introducing the group train member.
Di ba't sabi mo, napaka-onyx ko? Huwag nang magpaganda, sayang lang ang oras ko. Sa kapal ng kilay, pwedeng hide out. Sa itim ng balat, yari pag nag-brown out. Lumipas ang ilang.
So, lesson 7, nucleic acids. So, there are 4 learning outcomes. So, first, identify the components of nucleic acids and nucleotides. Second, distinguish between DNA structure and RNA structure. Third, list down the steps in DNA replication. And the fourth is describe the kinds of RNA. So, let's start. Nucleic acids. So nucleic acid. So in this image here, we will talk about the nucleic acid and the transfer of genetic information to new cell. So nucleic acids are biomolecules that transfer genetic information to new cell, and you know what? There are two types of nucleic acid. So here. So the RNA, or the what we call ribonucleic acid, and the DNA, the deoxyribonucleic acid. So RNA is found mainly in the cytoplasm of living cell. So DNA, it is found mainly in the nucleus of living cell. That RNA and DNA are the two polymers, which means that they have made up of repeating subunits called nucleotides. You know what's the 3 components of nucleotides? So, first is the sugar, or we call ribose or deoxyribose. Second, the phosphate. The third also is the nitrogenous base or bases, and other term of purine or a pyrimidine. So heterocyclic bases, these are the nitrogen containing molecules that come in the varieties of purine and pyrimidines. So second, the sugar. Sugar is a simple carbohydrates that provides energy for the cell, and the type of sugar in nucleotides determine whether it is RNA or DNA. And the third is the phosphate group. So this group of atom contain phosphorus and also phosphate. So this group link nucleotides together to form a chain. So heterocyclic bases, a ring that contains elements other than carbon, is called a heterocyclic ring. So the bases found in RNA and DNA contain 2 types of heterocyclic rings, which is the pyrimidine. So this is pyrimidine sample, and the purine. So pyrimidine has a has 6 single 6-membered rings. So 1, 2, 3, 4, 5, 6. So 6-membered ring and has a single. So this is the single. And in the purine, we have also it is a double ring structure. So double 1, 2. So this is the structure, and first structure we have 6. So 1, 2, 3, 4, 5, 6. And the second structure we have 5. 1, 2, 3, 4, and 5. So pyrimidine, we have uracil, thymine, and cytosine. So in purine, we have adenine, guanine, and it is important to know that DNA contains adenine, guanine, thymine, and cytosine. And RNA replaces thymine and uracil. So this is the example of heterocyclic bases. In pyrimidines, we have uracil, thymine, and cytosine. So pyrimidine, it has 6 single membered rings. So 6. 1, 2, 3, 4, 5, 6. And also the thymine and cytosine. So this uracil only in RNA and thymine only in DNA. And this tree is the example of pyrimidines that has 6 single membered ring. So in purine, as I said a while ago, that it is a double ring structure and with 6 member fused to 5 membered ring. So from 6 to 5. So the example of this is the adenine and the guanine. So these are the examples of heterocyclic bases in pyrimidines and purine. So sugars and phosphate group. So in RNA, the sugar component is the ribose, and in DNA, the sugar is deoxyribose. And note that both sugars are in the beta anomeric form. So first example is the B-derivose. So we have 5 carbon. So carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. The role of carbon 1 is the nitrogenous base will stick ATGC or the adenine, thymine, guanine, and cytosine. So here in carbon 2, this carbon 2 has no oxygen. That's why we call deoxyribose instead of oxyribose. Deoxyribose means no oxygen, only hydrogen. And the third carbon, in the third carbon, this part is the hydroxyl group. It helps to connect us to the next nucleotide. So in carbon 4, the ring carbon is created here. This is the ring carbon because as we can see, this carbon 4 and carbon 1 are connected by the ring structure. And the carbon 5 here, it is the phosphate group stick here. So this is the carbon 5 phosphate group will stick. This is the example of deoxyribose. So both have 5 carbon, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. But the difference is in carbon 2, there is no hydroxyl group in deoxyribose. So the phosphate group in nucleotides is derived from phosphoric acid H3PO4 and the psychological pH exists in the iconic form. So this is the example, and this is the double bond, this is the oxygen, hydroxyl group, oxygen, and it is negative, and also oxygen, and it is same negative. So we have two negative oxygen, two double bond, and one hydroxyl group.
So, good day everyone. I am Jason Gimonopolio, 20 years old, taking Bachelor Science in Agriculture, second year. So, I am the member of Group 3. So, for this video recorded, we will continuing the my reporting. My assigned topic, it's all about the nucleotides and the general structure of nucleotides. So before with that, uh, I can say that thank you to our instructor Mamel Dizon for the very kindness instructor. Also to my groups. So now we will for this video recorded, we will discuss about the nucleotides. So nucleotides are molecules that make up nucleic acid, so, and clicking and clacking together to form DNA and RNA. So nucleotides are made of three main components: E5 carbon sugar molecules, that is a ribose for RNA, and the deoxyribose for DNA. The other two components of nucleotides are phosphate group and nitrogen base called a nucleic base. So nucleic acids are components of a nucleotide chains linked by 3 and 5 phosphodiester linkage. So a phosphodiester linkage or bond, you can also call in a bond, combine the phosphate group and the hydroxyl group. So there are two sugar through a condensation reaction. The third carbon of one sugar joins with the 5 carbons of another and voila. So nucleic base that make up nucleotides and pair together to form DNA and RNA. So that you have adenine, cytosine, guanine, thymine, and uracil, and they group together in complementary ways to form nucleic acid, that is RNA. So in DNA, they have adenine and thymine grouped together with cytosine and guanine group and also grouped together. So in RNA, uracil replaces thymine, so the group pairing to adenine and uracil, and cytosine and guanine nucleotides. We will move on the nucleotides. We will go to the nucleotide, the general structure of nucleotide. So as you can see the structure below, the general structure of nucleotides consists of three main components. So that is a nitrogenous base, a 5 carbon sugar, and phosphate group. The nitrogenous base can be adenine, guanine, cytosine, or thymine. So that's a nitrogenous base belong to the adenine, guanine, cytosine, and or thymine. In DNA or uracil in RNA, the sugar is either ribose in RNA or deoxyribose in DNA. The phosphate group links the sugar of adjacent nucleotides together, forming the backbones of the nucleic acid chain. Together, these components create the basic building blocks of DNA and RNA, essential for storing and transmitting genetic information. So, so the nitrogenous base, there are two categories of nitrogenous base. This this is purines and pyrimidines. This the two the two categories of nitrogenous base are purines and pyrimidines. The adenine and guanine are purines, characterized by the double ring structure, while cytosine, thymine, and uracil are pyrimidines with a single ring structure. So, ang cytosine, there are two double ring structure, and the pyrimidines is a single ring structure. In DNA, adenine pairs with a thymine, guanine pairs with cytosine, and forming complementary base pairs. In RNA, thymine is replaced by uracil, so adenine pairs with uracil. And the second is sugar. The sugar components of nucleotide is either ribose or deoxyribose. The ribose and hydroxyl group attached to the second carbon's atom, while deoxyribose lack this group. The presence or the absence of this hydroxyl group differentiate ribonucleotides found in RNA from the deoxyribonucleotides found in DNA. 5 carbon atom of one sugar molecules to the 3 carbon atom of next sugar molecules in the chain, forming a sugar phosphate backbone. So this linkage occurs through a phosphodiester bonds. And another explanation, if the combination of these components forms nucleotides, and when nucleotides join together via phosphodiester bonds, they create nucleic acid chains such as DNA and RNA. These chains serve as the foundation for the storage and transmission of genetic information in living organisms. So that's all my assigned topic, and that's all, and thank you.
Hi guys, I'm Lorraine Diongkodoran. So, I'll be reporting primary and secondary structure of a DNA. Primary structure of DNA. DNA is one of the largest molecules known, containing between 1 and 100 million nucleotide units. DNA, or deoxyribonucleic acid, is indeed a large molecule. It composed of a sugar molecule, deoxyribose, a phosphate group, and one of 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. The sequence of these bases along the DNA strand forms the genetic code, which contains the instructions for the development, functioning, growth, and reproduction of all living organisms. DNA is responsible for storing and transmitting genetic information from one generation to the next through the process of replication and cell division. The nucleotides in DNA are linked by phosphate groups that connect the 5 carbon of one nucleotide to the 3 carbon of the next. Because these connections occur on two oxygen atoms of the phosphate group, they are called phosphodiester bonds. A phosphodiester bond is a chemical bond that holds together the backbone of DNA and RNA molecules. It forms between a phosphate group and 2 adjacent hydroxyl groups of pentose sugars, creating a chain-like structure. This bond is crucial for the stability and integrity of nucleic acid molecules. The nucleic acid backbone then is a sequence of sugar phosphate groups, which differ only in the sequence of bases attached to the sugars along the backbone, or the primary structure of DNA. So below is the primary structure of DNA. So this illustration shows the phosphodiester bonds. It consists it consists nitrogenous base and pentose sugar and phosphate group. Next is the second structure of DNA. The bases hydrogen bond to each other in a specific way. A hydrogen bonds to T and G hydrogen bonds to C, forming a set of complementary base pairs. Complementary base pairing refers to the specific hydrogen bonding between nitrogenous bases in DNA. Adenine pairs with thymine and cytosine pairs with guanine. This complementary base pairing ensures that the two strands of DNA can accurately replicate during cell division, as the sequence of one strand determines the sequence of the other. This principle is also important in techniques like PCR or polymerase chain reaction and DNA sequencing, where knowing the complementary base pairs allows scientists to amplify and study regions of DNA. This allows two separate strands of sugar phosphate backbones to run alongside to each other, held together by the hydrogen bonds between the complementary base pairs. So adenine paired with thymine and thymine paired with adenine and cytosine paired with guanine and guanine paired to cytosine. So that's all and thank you.
Hello everyone! My name is Rachel Maata Martin, and for today's video, I'm going to report about the double helix. So imagine a microscopic dance party. Two long, elegant sugar phosphate chain twist and group. Their rhythm controlled by funky colored partners clinging to them. So these partners aren't just there for fun. So they're the A team or the adenine, G squad for guanine, C crew for cytosine, and T troop for thymine. So nitrogenous bases that hold the key to life's biggest secrets. This, my friends, is the double helix, the iconic structure of the DNA. So get ready to untangle the mystery behind this microscopic masterpiece. Well, delve into how scientists crack the code, the funky moves of the bases, and why the discovery is the ultimate party favor for understanding life on Earth. Buckle up, because we're about to get schooled by the coolest molecule of the DNA. The two strands that twist around each other in a helix like shape represent the DNA molecule. Each strand is made up of units called nucleotides, which consist of sugar, a phosphate group, and a nitrogenous bases. So the image identifies the four different nitrogenous bases found in DNA. So A for adenine, G guanine, C for cytosine, and T for thymine. The two strands of DNA are held together by hydrogen bonds between the nitrogenous bases. So adenine always pairs with thymine and the guanine pairs with cytosine. So these specific base pairs are what cause the double helix shape. Antiparallel strands. The two strands that make up the DNA molecule run in opposite direction. So this is referred to as an antiparallel. One strand runs in the 5 to 3 direction, So the sugar phosphate backbone, which forms the outer edges of the helix, contains alternating sugar and phosphate groups. So the sugar group is always diacylribose in DNA. The phosphate group attaches to the hydroxyl group, or OH, on the fifth carbon atom of the sugar in one strand and the third carbon atom of the sugar in the other strand, linking the two strands together. So, in conclusion, this depicts the double helix structure of DNA, which is essential for carrying genetic information. And that's all for my report. Thank you.
Good day everyone, my name is Kirby Secon and I am the next reporter. So, my report is discovery of DNA structure. DNA was discovered in 1869 by the Swiss physician Friedrich Miescher in the post of discarded surgical bandages. He named it nuclein because it was located in the nucleus of the cell. Pus is a thick yellowish or greenish fluid that forms when the body fights off infection. Friedrich Miescher first isolated a substance from a nucleic of a white blood cells in post-soaked bandages. He named it nuclein, which we know today as DNA. In 1878, Albrecht Kossel isolated the pure nucleic acid and later isolated the 5 nitrogenous bases. So the 5 nitrogenous bases are adenine, thymine, cytosine, guanine, and uracil. Many scientists believe that nucleic acids were far too simple to be agents that carried genetic information from one generation to the next, and that the genetic material would turn out to be protein. So in 1943, Oswald Avery, Colin MacLeod, and Maclean McCarthy identified DNA as a carrier of genetic information. The race was on to determine the structure of DNA and how it was able to transmit genetic information. So the scientific community raced to determine the structure of DNA and how it carried genetic information. So this led to one of the most significant discovery in biology by James Watson and Francis Crick in 1953 when they proposed the double helix structure of DNA. In 1952, Rosalind Franklin obtained an X-ray crystal structure of a sample of DNA which contained structural features which led James D. Watson and Francis H. C. Crick to deduce the double helix structure of DNA. So in 1952, Rosalind Franklin took a special photo of DNA using X-rays. This photo showed that DNA looked like a twisted ladder, which was a big clue. So James Watson and Francis Crick used Franklin's photo to figure out that DNA is shaped like a double helix, like a spiral staircase with two sides. The diagram shows us how Watson and Crick thought DNA was structured. The two ribbons represent the sides of the ladder, and the rods between them represent the steps of the ladder, made of pairs of chemical bases. These chemical bases are adenine paired with thymine and guanine paired with cytosine, also called deoxyribonucleic acid. We wish to suggest a structure for the salt deoxyribonucleic acid, DNA. This structure has novel features which are of considerable biological interest. It does not escape our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So, Watson and Crick's proposed the double helix structure of DNA. They noted the unique pairing of nucleotide bases in their model, which suggested a mechanism for DNA replication, a crucial process in genetics. So, that is all for my report.
Good day, everyone, especially to our biochemistry instructor, Ma'am Meljon B. Dizon. And I am Ramon Binaro Vilutindos, one of the member of the Group 3. And I am tasked to report about the DNA replication. So, allow me to use my screen so that you can see what I am reporting or showing in the screen. So, okay, so DNA replication. So, let's start in chromosomes. So, by the way, what is chromosomes? So, the chromosomes are the thread-like structure as you seen in the screen. So, It is located inside the nucleus of animal and plant cell. Each chromosome is made up of protein and single molecule of deoxyribonucleic acid or the DNA, passed from parents to offspring, or the DNA contains the specific instruction that make each type of living creature unique. So how many chromosomes do human have? So we have 23 pairs of chromosomes, X and Y chromosomes of a total of 46, each of which chromosomes contains molecule of DNA coiled tightly around a group of small basic proteins called the histones. So as you see in the screen, so uh, I'll zoom it. So here in the lower part of the screen, there's something like a thread-like structure. There's a small round thing there, so that is called the basic or the small basic proteins called the histone, which the chromosomes are the DNA that was coiled up tightly. So there. Okay, so what do chromosomes do, by the way? So the unique structure of chromosomes keeps DNA tightly wrapped around spool-like proteins called the histones. So as I said, without such packaging, the DNA molecules would be too long to fit inside the cells. For example, if the DNA molecules in a single human cell were unwound from their histone and placed end to end, they would stretch 6 feet. So, as you can see, something like it was a thread that was circled. So, if that is get wound, so it will stretch up. So, for an organism to grow and function properly, cell must constantly divide to produce um, new cells to replace old worn out cells. During cell division, it is essentially that DNA remains intact and evenly distributed among cells. So chromosomes are key part of the process that ensures DNA is accurately copied and distributed in the vast majority of cell division. Still, mistakes do occur on rare occasion, but that is just rare. So it was accurate to in a process that ensures the DNA will accurately copied. So that's why it is called replication, because of the DNA was duplicated and ensures to have the two daughter DNA. So two daughter DNA that was um what do you call this? It is so the DNA is copied correctly. Changes in the number of structure of chromosomes in new cells may lead to serious problem. For example, in human, one type of leukemia and some other cancers are caused by defective chromosomes made up of joined pieces or broken chromosomes. It is also crucial that reproductive cells, such as eggs and sperm, contain the right number of chromosomes, and that those chromosomes have resulting offspring may fail to develop properly. For example, people with Down syndrome have three copies of chromosomes instead of two copies found in people. Okay, so that's it. So next is, okay, so kindly look at the screen. So there's a picture there, the image of the chromosomes, the cells. So okay, so here, so the chromosomes is located inside the nucleus in the cell. So as you can see there, the okay, I will zoom it. So in chromosomes, so there we have uh, the centromeres, the centromere, telomere, and chromatid. So what are those things in the chromosomes? So let's start in the centromere. So this constricted region of linear chromosomes is known as the centromere. So what is found there in the screen. So although this constriction is called the centromere, it is usually not located exactly in the center. Uh, as I said a while ago, that the this centromere is found inside or the center of the cell, but it is not usually located exactly. And in some cases, it is located almost at the chromosome's end. So there is just it is found here in the um, located in the end or in the bottom. The region on either side of centromere are referred to as the chromosome's arms. So it is said that these regions may also referred as chromosome's arms. So because it is located here top by the way, so it's like an arm. So centromeres help to keep chromosomes properly aligned during the complex process of cell division. As chromosomes are copied in preparation for production of new cell, the centromere serves as an attachment site for the two halves of each replicated chromosomes known as the sister chromatids. And in the other hand, the telomeres are repetitive stretches of DNA located at the end of the linear chromosome. So it is located here. As you see in the screen, it is located in the bottom. So they protect the ends of the chromosomes in a manner similar to the way the tips of shoelace keep them from unrevealing. In many types of cells, telomeres lose a bit of their DNA every time a cell divides. So eventually, when all of the telomere DNA is gone, the cell cannot replicate and dies. So it cannot replicate. So it eventually, if it is gone, white blood cells and other cell types now with the capacity to divide very frequently have a special enzyme that prevents their chromosomes from losing their telomere because they retain their telomeres. Such cells generally live longer than other cells. So these telomeres also play a role in the cancer. So the chromosomes of malignant cells usually do not lose their telomeres, helping to fuel the uncontrolled growth that makes cancer so devastating. Okay, so let's proceed to the next slide. Okay, so next is genes. So, individual section of DNA molecules make up the genes, which are the fundamental units of heredity that direct the synthesis of proteins. So, for example, example, in humans and most other complex organisms, one copy of each chromosome is inherited from the female parent and other from the male parents. This explains why children inherit mother and father's DNA. So the pattern of inheritance is different from the small circular chromosomes found in mitochondria. Only egg cells and not sperm cells keep their mitochondria during fertilization. So mitochondrial DNA is always inherited from the female parent. So, in humans, a few conditions, including some forms of hearing impairment and diabetes, have been associated with DNA found in mitochondria. So, what do characteristics of the male or in the female? So, it will be inherit, or some other diseases would be inherit to the offspring. So, that's why there are um, abnormalities we acquire or some children's acquire. It is because it inherit from their parents. So here we will know here how many genes do cells or the virus has. So here viruses contains a few to several hundred genes. So the virus only have a few hundreds of genes. But on the other hand, E. coli virus or the Escherichia coli contains approximately 1,000 genes. So it is greater than the other virus. But moreover, human cells contains approximately 25,000 genes. So imagine, so it's a lot of genes do the human cells have. So that's why those genes will pass through from parents to the offspring. Okay, so now let's proceed to replication, the main point of my report. So what is replication, by the way? So here, the definition. So replication is the process by which an exact copy of DNA is produced. So two strands of DNA separate and each one serve as the template for the construction of its own complement, generating new DNA strands that are exactly replicas of the original molecule. So for example, in a double helix structure of the DNA will divide, so the the other side of the helix will have the template of the exact DNA molecule. So um, it will be the template for in order for to produce another DNA. So there is we have the molecule called the adenine, thymine, and the so there we have the template of the exact sequence of the molecule. And then this is now called the daughter DNA molecule. So these two daughter DNA molecule have exactly the same base sequence of the parent's DNA. Each daughter contains one strand of the parent and one new strand that is complementary to the parent strand. This type of replication is called simic conservative replication. Okay, so as you can see in the screen, so here, the DNA replication. Okay, so as you can see in the screen, there we have the original parent DNA. So the first strand have the adenine, thymine, adenine, then cytosine, cytosine to adenine. So adenine is always paired to thymine. Then it can be also interchanged, thymine to adenine, adenine to thymine. And another one is the cytosine to guanine, and they should not be interchanged to the other molecules too. So in the first strand, as you can see there, then if the original parent DNA, if that divides, so we have the two replicas of the two daughter DNA molecule. So there, if the other one, the other DNA that consist of the same replicas or the same sequence, so the okay, so in the original DNA, so the other part, we have the first strand, so we have the sequence of adenine, thymine, adenine, cytosine, cytosine, two cytosines, and adenine. So one daughter DNA molecule, this still have the similar sequence of the strand. So we have there adenine, thymine, adenine, two cytosines, and adenine. So that is exactly the same as the original parent strand. So in the other side of the other strand of first strand of the DNA, and the other daughter DNA molecule also have the other strand, which is exactly similar to the second strand, which is the thymine, adenine, thymine, guanine, guanine, and thymine. So that is called replication. So thank you so much and good day.
Hey guys, I am John Christian Siabuhan, and the topic that I am going to discuss is all about DNA replication. So, life of a cell or cell cycle. So, it is actively preparing to divide into two daughter cells. The cell cycle starts at the interface, the G1, S, and G2, which cells prepare for division. So, Then mitosis, which the cell divides. Then the S phase, which cells performs DNA replications. So that the 46 chromosomes are duplicated. So take note that a single chromosome is made up of a single DNA molecule that has two strands forming a double helix. And each single-stranded DNA is composed of four types of nucleotides, which are the building blocks of DNA. The nucleotides of one strand from the hydrogen bond to the complementary nucleotides on the other strand, specifically thymine bond with adenine and guanine with cytosine. So the strand of DNA run from 3' to 5', while the other runs from 5' to 3'. And murag duha ka sawa that coils up together but facing a different direction. So now let's discuss DNA replication. It can be described as semi-conservative, which means that the strands of the double helix act as a template based on which complementary standards will form, which eventually the original chromosomes will split into two exact copies, each made up of one old strand from parent DNA and new daughter cells strand from the new strand. So overall, DNA replication has a step: the initiation or unwinding of the double helix, the elongation or the synthesis of DNA segments, and lastly, termination or closing the neck. So, initiation kicks off when a group of protein gets together to form the pre-replication complex, where it looks for specific sequence in DNA strand called the origin of replication. So take note, the enzyme DNA helicase catalyzed the separation and unwinding of the nucleic acid that stands, which created a replication fork. So the segment ahead of it starts to overwind, meaning the double helix is tightly wind. So the DNA topoisomers works ahead of the helicase to loosen up the tightened DNA coils. Now, next step is elongation. So here, we need the RNA primase. Its function is to hover around in randomly synthesized small lengths of RNA called RNA primers. And just by chance, the RNA primer will complementary to the exposed DNA strand and it will bind. Then, the enzyme RNA polymerase will latch on the short double-stranded segments and add more complementary nucleotides on the template strand into a 3' to 5' direction and is called a leading strand. And the other strand is from 5' to 3' and called the lagging strand. So for the other templates, strand that runs from 5' to 3', it is complicated because of the antiparallel. So to solve the issue, the RNA primase lays down a number of primers that binds to different spots along the length of the DNA template strand, which offers a lot of 3' ends. So the DNA polymerase can then add nucleotides to the 3' end of each of the primers and is called the Okazaki fragments. So these fragments are joined by the enzyme called DNA ligase, resulting the finished strand of called the DNA called the lagging strand. Now, for the final step, termination or closing the neck. Miss Edeline Ibaw will continue.
So hello everyone, this is the continuation of steps in DNA replication. So let's go. After synthesizing of DNA segments, the next step is closing the nicks. What is nicks? So nicks is the gaps or breaks between segments and the daughter strand for the smooth running of DNA replication. The Okazaki fragments are the DNA fragments being separated by the nicks. To furthermore understand the step 3, the closing of the nicks. So the daughter strand along the leading strand is synthesized smoothly without any nicks. So the Okazaki fragments along the lagging strand that are joined by an enzyme that are called DNA ligase, which is removes the DNA primer and replaces it with the correct nucleotides. The result of the two DNA double helix molecule of DNA are identical to the original DNA molecule, each of which contains one old strand from the parent DNA and the new daughter strand. As we see in the structure of a DNA replication, we can see the three different steps in DNA replication, which is the first one. The DNA is unwound by helicase and a replication fork forms. After that, with the help of the enzyme DNA polymerase, we can see in the structure that DNA primase. So for step 2, DNA is replicated smoothly along the leading strand, which grows towards the replication fork. So the DNA, or the what we call Okazaki fragments, are synthesized by DNA polymerase along the lagging strand as the replication fork moves. For step 3, the Okazaki fragments are joined by DNA ligase. This results in two new DNA molecules, which we can see in the left side. So DNA replication in more than one place. In eukaryotes, DNA replication occurs simultaneously at many replication forks along the original molecule. The zones where replication occur eventually combined to form complete strand. This allows the long molecule to be replicated quickly. So in the fruit fly, the largest chromosomes of fruit fly would take more than 16 days to replicate one segment. The actual process takes only less than 3 minutes because it occurs more than 6,000 replication forks. So we have here some example of DNA replication in more than one place as follow. So here is the structure of DNA replication in more than one place, which we can see the parent DNA and it follows the replication occurring at several points, and after that we can have the daughter DNA.
Good day, Ma'am Mel. Good day, everyone. My name is Milani El Amoso, the next reporter. So my topic is all about polymerase chain reaction. First, what is polymerase chain reaction? The polymerase chain reaction, or PCR, is a fundamental laboratory technique used to amplify a specific segment of DNA. It involves a series of temperature cycles that replicate the targeted DNA sequence, making it possible to produce millions of copies of, of that particular segment for various applications in research, diagnostic, forensic, and in other fields. An important laboratory technique called the polymerase chain reaction, or PCR, mimics the natural process of replication. A small quantity of target DNA, a buffered solution of DNA polymerase, the cofactor of magnesium and chlorine, the four nucleotide building blocks and primers are added to the test tube. The primers are short polynucleotides that bind to the DNA strands and serve as the starting points for a new chain growth. The mixture goes through several three steps of replication cycles. The polymerase chain reaction, or PCR, works by repeatedly heating and cooling a reaction mixture containing DNA primers, or the short DNA sequence that bind to the target DNA, nucleotides, building blocks of DNA, and a DNA polymerase enzyme. The several three steps of replications are: first, the process typically involves three main types. These are the several steps. First, the denaturation. Second, annealing. Third, extension. So first, denaturation. The reaction mixture is heated to a high temperature, or the heat is 94 to 96 degrees Celsius, is used for several minutes to unravel DNA into single strands, causing the double-stranded DNA to separate into two single strands. Next, the annealing. Temperature is lowered to allow the primers to bend to the complementary sequences on each of the single DNA strands. The tube is cooled to 50 to 65 degrees Celsius for to several minutes to allow primers to hydrogen bond to the separated strands of target DNA. Next, the third one, extension. The temperature is raised again, and the DNA polymerase enzyme extends the primers by adding nucleotides to synthesize new DNA strands complementary to the target sequence. The tube is heated to 72 degrees Celsius for to several minutes while DNA polymerase synthesizes new strand. These three steps are repeated in multiple cycles, usually 20 to 40 cycles, to exponentially amplify the targeted DNA region, resulting in a significant increase in the amount of the specific DNA segment. The amplified DNA can be analyzed, sequenced, or used for various applications in research and diagnostic. Each cycle doubles the amount of DNA. Following 30 cycles, a neoretically amplification factor of 1 billion attained. PCR is a standard research technique that detects all manners of mutations associated with genetic disease. It is also used to detect presence of unwanted DNA or the bacterial or viral infection. It's a fast, simple alternative to lengthy procedures involving sample cultures that can take weeks. It can also be used on degraded DNA samples like forensic analysis, DNA fingerprinting, recovery of DNA from extinct mammals, Egyptian mummies, and ancient insects trapped in amber to be amplified and analyzed. It offers several key advantages and applications, including amplifying DNA, DNA cloning, genetic testing, drug development, evolutionary studies, microbial detection. Overall, PCR is a versatile and powerful tool that has significantly advanced research in genetics, biotechnology, medicine, forensics, and many other scientific disciplines. Thank you.
Good afternoon, ma'am. My name is Angeline Amy Harris, Section A. Linoleic acid RNA is a butyl molecule present in cells that plays a circular role in various biology. Process RNA is a long unbranched polymer composed of nucleotides link together by phosphodiester bonds in a 3 to 5 direction. These are RNA strand can vary in length from 73 nucleotides to many thousand. Unlike DNA, which is primarily located in the nucleus of cells, RNA is found in different parts of the cell, including the nucleus, cytoplasm, and mitochondria. Ordinary. One of the key differences between RNA and DNA lies in their primary structures. In RNA, the sugar component is ribose, while DNA contains deoxyribose. Additionally, RNA uses the base uracil instead of thymine, which is specific to DNA. This distinction in sugar and base components between RNA and DNA contributes to their unique functions between the cell. The secondary structure is of RNA refers to the local folded structure within an RNA molecules, including regions where the RNA forms double helical structure and loops. In RNA, base pairing occurs between adenine and uracil as well as between guanine and cytosine, forming stable base pairs. In the sequences these sequences contains a series of bases where guanine is paired with cytosine and adenine is paired with uracil. These sequences consist of three uracil bases which may form a loop or single-stranded region. In the RNA structure is here cytosine is paired with guanine and adenine is paired with uracil. The base pairing roles in RNA is these sequences show a single base pair between guanine and cytosine. These base pairing and sequences contribute to the secondary structure of RNA molecule, allowing it to fold into specific shape and functional structure and necessary for its biological function. And
Hi guys, this is Barbie J. Salak and my topic is all about kinds of RNA. So, there are three major types of RNA which occur in all organisms. There are the messenger RNA that is mRNA, the ribosomal RNA that is rRNA, and transfer RNA that is tRNA. So, before that, let's tackle the messenger RNA or the mRNA. So, messenger RNA is a function as a carrier of genetic information from the DNA in the cell nucleus to the site of protein synthesis in the cytoplasm. The bases of mRNA are in complementary sequence to the base sequence of one of the strands of nuclear DNA. mRNA has a short lifetime. Usually less than 1 hour, it is synthesized as it is needed rapidly degraded to the constituent nucleotides. So, messenger RNA, as the name suggests, mRNA comes the genetic information from DNA to the ribosome's genetic. And the DNA are transcribed into the mRNA by a process called the transcription. Here the message is translated into action that is based on the genetic information. Different types of proteins are synthesized. The type of gene that is involved in the protein synthesis depends upon the strand, kinds, and sequence of nucleotide. It is about 3 to 5% of the RNA content of the cell. The mRNA is always single-stranded. The mRNA is produced as complementary copy of the DNA which is involved in the protein synthesis. So that's that is for the messenger RNA. Let's move to the ribosomal RNA or rRNA. The main component of ribosomes that are the site of protein synthesis. rRNA accounts for 80 to 85% of the total RNA of the cell. rRNA accounts for 65% of a ribosome structure. The remaining 35% is protein. So, ribosomal RNA that is found in the ribosomes, the rRNA represents about 40 to 60% of the total weight of the ribosomes. Relatively, it consists about 80% of the total RNA of the cells. They are produced in the nucleus. They are, they are the most stable forms of RNA. They consist of single strand of nucleotides. At some region, the strand is folded. So that is for the ribosomal RNA. So we are now at the transfer RNA or tRNA. tRNA delivers individual amino acids to the site of protein synthesis. tRNA is specific to one type of amino acid. Cells contain at least one specific type of tRNA for each of the 20 common amino acids. tRNA is the smallest of the nucleic acid with 73 to 93 nucleotides per chain. So this is the table of different forms of RNA molecules in Escherichia coli or E. coli. So we have the class of RNA present in cells, number of RNA subtypes, number of nucleotides. For the class of RNA, we have the ribosomal RNA or rRNA, transfer RNA or tRNA, and messenger RNA or mRNA. So for the ribosomal RNA or rRNA, we have 80%. Number of RNA subtypes, we have 3. Number of nucleotides, we have 120 to 1,700 and 3,700. For the transfer RNA or tRNA, we have 1.5% in cells. Number of RNA subtypes, we have 46. Number of nucleotides, we have 73 to 93. And for the messenger RNA or mRNA, we have 5%. And the number of the RNA subtypes, many. Number of nucleotides, we have 75 to 3,000. So next, we have the transfer RNA. tRNA has region of hydrogen bonding between complementary base pairs separated by loops where there is no hydrogen bonding. Two regions of RNA have important function. The anticodon is 3-base sequence which allows tRNA to bind to mRNA during protein synthesis. It is complementary to one of the codons in mRNA. The 3 end of the molecules binds an amino acid with an ester bond and transport into the site of protein synthesis. An enzyme matches the tRNA molecules to the correct amino acid, activating it for the protein synthesis. So, let us know about the transfer RNA. Transfer RNA is also known as soluble RNA or sRNA. The tRNA is a small molecule compared with other types of RNAs. It amounts to about 15% of total RNA of the cell. The tRNA molecules performs a number of functions. The most important one is to act as a carrier of amino acid to the site of protein synthesis. So, there are about more than 20 types of tRNAs. Each tRNA is specific for a particular amino acid. In bacterial cell, there are more than 70 tRNAs, and in eukaryotic cell, the number of even greater. There are 4 or 5 tRNAs specific for a particular amino acid, and this is called isoacceptor tRNA. So, as you have seen the picture, you can see the site of amino acid attachment down to the anticodon site of attachment to mRNA. So this is called the transfer RNA, and you have seen the activated tRNA and the activated tRNA schematic. So that's all for my report, and thank you.